Welcome to NewYorkBIO's virtual breakfast series. The digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode, we feature serial entrepreneur and company creator Michael Gilman.
Yeah. All right. Good morning, everyone. My name is Jennifer Hawks-Bland, and I'm the CEO at NewYorkBIO. We are very excited you're here to hear our conversation with Michael Gilman today. A special thank you to our sponsor, Pfizer, for this month's series of breakfast. And with that, I almost always forget this. You think I should have it on a recording and push the button. During our conversation today, please ask your questions either in the chat or the Q&A feature. And Derek and I will get to them throughout the course of our conversation. We're very happy to have Mike joining us, and I'll kick it over to Derek to start our conversation. Thanks.
All right, Mike, good morning. Thank you so much for being here. Honestly, I could just about start it at any point in your career for kicking off kind of an intro. You were an academic scientist, you were in larger biopharma, you were at Ariad, Biogen, and have now started and exited multiple companies. So we usually tend to kick this off with an origin story, and you can kind of pick any point within your career to start. Why don't you tell us how you got where you are now?
Yeah, can't hold a job, right? Yeah, so I'm a scientist. I will always be a scientist. I've tried over the years to turn myself into a business guy, and it doesn't really take. I was a postdoc at the Whitehead Institute here in Cambridge, and then I actually took my first academic job in the New York area. I was on the staff at Cold Spring Harbor. So lived on Long Island for, I don't know, like 6 or 7 years. A couple of my kids were born there. And then moved to industry in 1994 when that was still a sort of a weird thing to do.
Yeah.
And I was doing okay at Cold Spring Harbor. I had was effectively a tenured position there. Like I could have stayed.
The rolling five.
Rolling five, exactly. Rolling five, which meant that, if on any given morning Watson woke up and decided to fire you, you had 5 minutes.
We lived on campus for a few months during the pandemic because my wife is a professor and she actually helps run one of the microscopy courses there. So we are now well versed in the rolling five politics and ups and downs of those things.
That's funny. I had not heard that term for a long time, but I was totally rolling five. So I actually uprooted, the family. We had three kids, single digit age at the time, and moved up here to take a job at Ariad Pharmaceuticals, which at the time was about 2 years old. And I would tell you, that was probably the most impulsive and ill-informed decision could have possibly. I had no idea what I was getting myself into, but it turned out that I loved it and I was really good at it. I'm not recommending impulsive and ill-informed decisions as sort of a career path, right? But what I am saying, for me, the actionable conclusion is that it pays to take chances, that, figuring out what you're good at, what makes you happy. I don't think you could, let me put it this way. I think very few people can impute that from first principles. It's sort of an experimental science. You kind of have to try stuff and figure out what sticks. And if something is not right, you usually figure it out pretty quickly and can typically unwind with minimal consequence. But I was lucky. It was really good. The thing that I really appreciated right away was 2 things. One was that, as an academic, what I realized was that I was, above all other things, a data junkie. I just loved getting new data and figuring out how things work. I was not so thrilled with the education mission. It's not that I didn't like it. It's just that I knew I was more interested in the data, right? And I found it sort of frustrating in a way, when I had a graduate student, working on an experiment that I just thought was really critical. And, she'd be struggling with it. And I knew like every fiber of my soul wanted to just like pick her up and put her in the hallway and just do the experiment myself, because I knew I could do it, right? But I also knew that it was important for her to figure out how to do it herself. And there were sort of two revelations for me once I moved to industry. One was that you actually didn't need to train people. If you needed an expert pharmacologist, you hired an expert pharmacologist, right? And number two is the fact that that science and industry is a team sport, and that there's sort of an alchemy to constructing effective teams. But once you have an effective and high functioning team, it is breathtaking the amount of data that they can generate. And man, I just took to that. And so I spent 5 years at Ariad. I got fired. That was the first of 3 times I got fired. And ended up at Biogen, and was at Biogen for, I don't know, 6 or 7 years, and eventually became the head of discovery research there. Went through the merger with IDEC, which I think was in 2003, which was experience. Got fired.
You want to talk about that, or do we not have enough time?
Well, I mean, there's a lot of things to talk about. I would think the main, I would think for me, the main lesson that I took away from that was that actually there was such a thing as corporate culture. I didn't really believe that was a thing until we had to merge these two organizations that were in fundamentally the same business, right?
Right.
But went about that business in completely different ways. I mean, it was astonishing, really. And I was actually on the integration team, and there was zero attention paid to addressing that cultural gap. And so, it devolved into, years of sort of pitched battles between the kind of the Cambridge people and the San Diego people, right?
Yeah.
Which it didn't have to be, I don't think. But anyhow, I mean, I could go on and on about that. But in some ways, I was a victim of that, and as being the only Cambridge-based executive who reported to Bill Rastetter, who was the chairman and CEO of IDEC, and was the executive chair of Biogen IDEC while Mullen was CEO. So anyhow, so I got fired from there in 2005, and that's when I kind of discovered my hidden entrepreneur. I was just out of there with no idea what to do with myself. And, but I started to, kind of connect with some of the local VCs, and quickly connected with the team at Atlas Venture, and spent, an interesting afternoon just brainstorming with them, things they were interested in, things I was interested in. And we left that meeting with this idea that we were going to start a company focused on fibrosis. That was sort of as the analysis had gone really at that point. And they're like, hey, we've got an empty office, figure it out, let us know. This was sort of before this sort of entrepreneur in residence and company creation was widespread. I mean, I was literally the only non-Atlas employee in the office. But it was exactly what I needed. Because at Biogen, I had just become a sad bureaucrat, honestly, right? I was running an organization of 400 people, and and although I love the science, the fact is I would come in the morning, I'd be issued my calendar card, and I would just roll from meeting to meeting all day long, right? It's more or less speaking when spoken to. And I would kind of come home at the end of the day, and I'm like, what the hell did I do today? And so the opportunity to sit by myself and think about a scientific problem and do my own research and read papers, and it reminded me why I was in this business in the first place. And it was just what I needed, and was quite invigorating. And we ended up starting a company. It was called Strometics. And we were able, in fact, to get an asset out of Biogen. It was a clinical stage antibody that obviously I was familiar with. that
Yeah.
Biogen had taken as far as IND and then basically dropped.
Shelved it.
And, you know, we were able to get it out of there, raised some money, got it to phase 2, at which point Biogen decided they'd like to have it back. And so, so they acquired the company. This was in 2012. So I've skipped over like 6 to 7 years that had some just glacious ups and downs, obviously.
You were not just sitting in the office at Atlas for those years, right?
Oh no, God,
you in-licensed the asset, 20 minutes passed, they bought it back. There you go.
Yeah. I mean, there was like I went through heart surgery in the middle of that. I mean, it's just it was like, I mean, man, oh man. But you know, what doesn't kill you makes you strong. And you know, bottom line, I actually went back to Biogen at a lower level than I left. So the whole thing was just a long, complicated and
7-year demotion.
Yeah.
Please let me work here again. I've taken one of your assets. I've advanced it. Can I please have any job?
Yeah, exactly. And so I lasted there a little over a year, got fired again. So I was number 3. So got fired twice from Biogen, which I can tell you it's actually pretty lucrative, right? Because, you know, but anyhow, that was great because I didn't really belong back there. Like I discovered what I'd been put on earth for, you know, and so I basically got liberated essentially. And I went back to Atlas and, you know, to sort of do it all over again. Although this time, now I realize I was no longer kind of like a subject matter expert. Like I was not going to produce the idea like I did. And so, you know, I just hung out in the office. I sat in there, you know, Monday morning meetings and kind of listened to all the things they were working on and kind of waited for sort of bells to go off in my head. And one day that happened. One day, Michael Gladstone, who I don't know if he's now a partner there.
He's now a partner, right?
He'd been working on, and it was a, you know, biology I'd never heard of before, but there was just something about it, you know, that just got my attention. I sort of raised my hand. I said, "Can I have that one, please?" And so I went to work on this, and this became my second company, which is called Padlock Therapeutics. And the idea there, Michael had been working with these 2 scientists at Scripps who were working on a family of enzymes called the PADs, peptidyl arginine deaminases. And the whole hypothesis there was that that maybe we'd gotten autoimmune disease all wrong all this time. That, you know, when we think of autoimmunity as a defect in the immune system where it gets confused and attacks, you know, healthy tissue, that it's not supposed to. But what if the immune system is actually just doing its job, and what it's reacting to is stuff it was actually never supposed to see, right? And you know, once you kind of reconfigure the problem of autoimmunity as it's not a problem of the immune system, but it's a problem of inappropriate production of immune and inflammatory material. You've got ways to think about treating the disease, and you don't have to mess with people's immune systems, which, you know, is not the best thing in the world to do. Anyhow, these PAD enzymes, peptidyl arginine deaminases, it's a mouthful, but they do something very simple, which is they post-translationally modify proteins on arginine residues to create a non-encoded amino acid called citrulline. And it turns out that in people with certain MHC backgrounds, HLA backgrounds, these citrulinated proteins are immunogenic. And for example, in rheumatoid arthritis, 75% of patients make antibodies to citrulinated proteins, and the presence of those antibodies is 98% specific for RA. So if you have those antibodies, it means you either have RA or you're going to get it. You can detect these antibodies in people like 10 years before they show up with clinical disease. So anyhow, so the idea was, well, you know, hell, you don't need to give these people Humira or whatever. You could treat them with a PAD inhibitor, and essentially break the inflammatory cycle. And the problem was that no one had succeeded in generating PAD inhibitors. It was it was a novel class of enzymes and all of that. And so we got to work on the chemistry. And I would just back up and say that the theme. I skipped many steps in the Stromedic story, but the emerging theme for me, the thing that I've probably learned as an entrepreneur that is probably the most important thing to sort of assimilate is that the way you create value, particularly in these sort of single asset companies or asset focused companies, is by really systematically discharging risk. And so that requires that you actually know where your risk is, which is not completely trivial.
Mm-hmm.
And then secondly, ensure that every experiment, certainly every clinical trial you do, is very specifically focused on taking some quantum of risk off the table. And coming back to Stromedics for a second, this sort of the killer app for this antibody was idiopathic pulmonary fibrosis, which is a very debilitating, fatal, progressive lung disease. And the problem at the time was that Biogen could not come up with a phase 2A trial design that would allow them to measure the clinical endpoints, which are the only endpoints they're really prepared to believe, right? And so they were looking at kind of going all the way to phase 3, which means probably spending $200-300 million, before you turn over the first card and go like, oh crap, I probably shouldn't have done that, right? And so and so the thing that we did at Stromedics, honestly, the one thing we did that created all that value for our shareholders and well, in theory for patients, although the program's dead now, was simply accepted that we could not test the clinical hypothesis in a relatively small phase 2 trial, but that we had a very strong biological hypothesis, which is that this antibody essentially inhibits TGF-beta activity in the lung. I could go deep into the weeds if anyone's interested. But the point is that we could measure quite statistically robustly in a small, short phase 2 for about 1/10 of the cost, right? And if the answer to that is no, that the biological hypothesis is false, then you're done. And you don't spend the rest of the money, right? But if it's correct, then you've taken a huge amount of the risk off the table. And really, Biogen, who had stayed close to the program, they had a board observer seat. Once they understood the trial that we had designed, and that we'd figured out a way to take out sort of 50% of the risk on 10% of the cost, that's when they made the offer to acquire the company. I mean, before we'd ever even dosed a patient in the trial. So there's a lesson for you, right? Like, I mean, we made money purely on the basis of a trial design, basically, right? And then in the case of Padlock, we found the biological hypothesis very compelling, but we quickly realized it's going to be very hard to prove because we figured out that the animal models were going to be useless, and that therefore, in order to prove that correlation is not causation, right? We would probably have to run a phase 2 trial in rheumatoid arthritis, and so we were kind of settled in, for that kind of 6 or 7 year run. But internally, we didn't see that as the main risk. We saw the risk as chemistry, right? We just didn't know if we were going to get chemistry against these targets. And if you can't get PAD inhibitors, then you don't start a PAD company, right? And so the whole seed investment, that 1 year of sort of part-time employment and outsourced chemistry, was all focused on trying to get some starting chemistry. And the idea was, if we didn't get it, we wouldn't even start the company, really. But if we did, we would. And we did. And we started the company. And it turns out we were right that chemistry was the proximal risk, because when we talked to a bunch of partners who were very interested in the hypothesis, And then we got to Bristol Myers Squibb, and they were like all over this biology. They totally believed it. They knew more about it than we did, clearly. But they had no chemistry. I mean, they never came out and said that, but I mean, it seemed clear they'd probably screened their corporate deck once or twice and not gotten anything. And we walked in there, honestly, like idiot savants with 18 months of chemistry against this target. And they're like, we have to have that, you know? And so they acquired the company in early 2016. I mean, the company was, I don't know, I mean, barely, I mean, it wasn't even 18 months old, had not spent its entire Series A. It was, you know, my complaint from my investors, because investors will always complain, right, was that they didn't have an opportunity to put more money in. You know, but too bad. And so this time I didn't really go back to BM. I was a BMS employee for 30 days, which, you know, was enough to get on their health insurance program, which was good. And then sorry, like I got to use the whole hour to answer your first question.
No, that's well, I was gonna, so I'll interrupt you slightly then, because in between, you know, if you think about kind of your initial foray into Stromedics, like, what was your learning curve like as an actual CEO? Like, what are some of the things that you took away from that immediately? And the other kind of piece I had in there was, what are some of the ways that the venture folks behind you were supportive, or in your mind kind of could have been more supportive? You know, what are some of the things that like would have been more helpful when you first started?
Yeah. Oh man, there's so many things that I learned in that experience. So I guess the first thing I would say is, once I was able to secure this clinical stage antibody from Biogen, which was 10 times better asset than any of the investors thought we would be able to get. One of the investors, who shall not be named, said, okay, now we've got to go get a real CEO. And I'm like, you know, and so I worked hard not to take that personally, right?
Yeah.
I mean, I took it very personally inside, but I tried not to let it show, you know? And I'm like, okay, explain to me exactly what this person would do, right? Because, you know, 80% of what this company needs to do over the next 2 years, I can do. Now, 20% I can't do, or at least I've never done before, right? And so, you know, I'm perfectly happy to admit that I need help on those things, but like you know, this company is about running the science, hiring the team, telling the story. Like I can do that shit, right? Like what, no, I mean, maybe you want somebody who's taller and better looking and all that sort of stuff. I get that, you know, but what would that person do? And, you know, so there was an interesting dialogue there. And, you know, at the end of the day, my friends at Atlas came through and they actually brought in Dan Lynch as executive chairman. So as, you know, Dan Lynch is like the quintessential executive chairman now, but this was the first time he did it actually. And he was, you know, he was kind of like my adult supervision, you know, a few years younger than me, but still a much more experienced, you know, business guy. And you know, and he was terrific, and he helped me a lot, and I learned a ton from him. And I, you know, and I think the thing I took away from that experience, like put immediately to work on Padlock, was realizing that it's okay not to know everything, right? That, you know, that, you know, for the longest time, I felt I was just so acutely aware of the things I didn't know as being flaws that I needed to remediate in some fashion. But, you know, what I now believe is like, there's some things I'm good at and there's some things I'm not good at, and that's just not going to change, right? And the fact is, you know, there are people out there who are great at the things I'm bad at, and I can hire them, right? I can hire them, you know, and they'll do a much better job of it than, you know, than I would ever do. And it was part of this transition for me which I think is challenging for anyone coming out of an academic background, that your job as a CEO is not to be a specialist, right? Your job is to be a generalist, right? The minute you know more about something that's going on in the company than anybody else, you're probably not doing your job properly, right? And that your job is to hold the whole picture in your head, and at some level sort of float above it all. Make sure you've got the right team and that team knows what it's doing. And one of the reasons I'm in the office today, like there's no work. I don't do any work that I can't do in my PJs at home, right? But I'm here because I realize an important part of my job is to just be around, just to kind of see and be seen. I mean, it's the funny job, the CEO job, right? You can't measure your success, at least on a day-to-day basis, the way you would in any other role, right? You can't count the number of widgets you made or whatever. So anyhow, I'm not sure I ever answered the question that you asked, but
I think you came close.
Yeah,
I think you came pretty close.
And we've actually had a question from the audience that I think fits here. The question is about your exits, right? And and the question is about the breakdown of upfront versus milestone, right? Payments as far as the acquisition goes. But take a step back. When you're negotiating for an acquisition of one of your companies, what do you think about from that, like milestone payments versus upfront? And how do you characterize the value when you're negotiating with another acquirer?
Well, I think you have to assume that the upfront is all you're ever going to get, because I just think, you have to settle a number that people will be happy with if that's all they're going to get. I think that, the whole sort of milestone structure or CVR structure is really about bridging the gap between, the valuation assumptions on either side of the transaction, right? Like, I think it's worth this much, you think it's worth this much. So, okay, we'll agree on some number closer to yours, but, as the thing becomes more like what we think it is, then we'll approach our value, right? And I think that's a reasonable, it's a reasonable conclusion. And I can tell you that in the case of Strometics, there was a substantial milestone payment that was larger than the upfront, right? So, so the employees, the shareholders, including the employees, actually did pretty well on that. In the case of Padlock, even the upfront there was, I don't know, probably like an 8x for the investors. On the basis of not a lot of money having been put in.
Yeah.
Yes.
So it was a big win for them, and milestones that get along the way are kind of gravy.
Yeah.
I'm just laughing at the fact that they were disappointed about that. It was, it's an 18-month exit, and there's still something to complain about. It's like, oh, we should have been playing longer. We should have taken this, and we should have, I'm sure someone somewhere had the thing. No, you reject this, and then we go here, and then we go back to them 6 months later, and then we try and do this, raise another huge thing, and yeah. Do you think? Well, so to just kind of stick on the investor front, right? So how much has changed from a funding perspective, right? If you think about where things were in 2012 was 10 years ago, right? And the investment environment has changed a lot, right? So are there kind of fundamental things that are different today, or is it still all kind of along the spectrum, and the pendulum will swing back at some point? Kind of, is all of this normal, or is anything from the investor side fundamentally changed?
That's a good question. At the end of the day, the market's fundamentally cyclical, right? And when the market gets hot, it overheats, and when the market gets cold, it goes into deep freeze, and it's really it responds irrationally in both directions, right? and so just speaking about today's market, people, I mean, the market today is, for small biotech companies is terrible, but it's only terrible if you need to raise money, right? If you don't need to raise money, you can, you could just wait for the market to cycle back, which it will do eventually. And, and here at Arrakis, I mean, we've been lucky. We've done a couple of significant partnership deals. We've got $250 million in the bank. And so, like, I don't need to be a market watcher today. I could sort of pick my spots. Strometics raised its first big round, it was a Series B, in 2008. And 2008 was really a market low.
Oh, yeah.
Industry, right? It was really, really terrible. But, I guess I would say two things. I would make two generalizations. Well, three, counting the markets are cyclical one. The second is that, kind of all politics is local. There's always money around, and if you've got a good story, you'll get it, right? You may not get the valuation that you want, but I also think that a lot of the debate about, valuation early on in the history of a company is sort of misplaced, because, startups are sort of binary events, right? They're either worth zero or they're a lot. And, I'm sure there are founders on this call who probably disagree with this, but at the end of the day, I'm going to say the valuation of your Series A round is going to wash out in the end. That's my view. And then the last generalization I would make is I do think that there is a lot more money around today than there was in 2008, and I think that's a permanent change. And I think it's a permanent change that's largely merited by the very substantial advances in our science. We are, as a discipline and as an industry, capable of things that people couldn't even conceptualize, 15 years ago. And so, I think there's inherently more value in what we do today. And as a consequence, I think there's, more investment capital available.
So we have a couple other questions, but I think this is a good time to ask this one. So the Arrakis story, you're drugging RNA, right? Which, I imagine when you first told people about that, they looked at you cross-eyed and had no idea what you were talking about, right? I mean, the science is going so far. How do you take a complex story like that and boil it down to something that people will, believe and get behind?
Yeah, well, so the reaction you described was actually my reaction to Jennifer Petter when she first told me that's what she was working on. Like, that seems crazy. But I knew Jen not to be crazy. And so I, spent quite a bit of time with her, listening to her ideas and thinking about them. And signed up as the CEO in October of 2016. I was employee number 2, Jen was employee number 1, and yeah, we hit the road to raise money in late 2016, early 2017, and that was a general reaction. Half the people we talked to said you're crazy, and the other half said you're brave, but you're crazy. And they all had a long list of reasons why it would never work, basically. Mm-hmm. And we thought pretty respectable theoretical answers for why it ought to work, but we didn't really have the data. But fortunately, we found some early believers. We were able to raise a decent Series A round, and we got to work. And we went right to work on the chemistry problem, because that was the one that people were focused on. You'll never get chemistry. You'll never get progressible chemistry against RNA. So that's where we focused our effort, and it took us a couple of years to figure out how to do it, but we did. And, and today we've got this, very efficient engine where, given an RNA of therapeutic interest, we can figure out where the pockets are, we can make those RNAs, we can recapitulate those folds, we can screen them, we can get chemistry, we can advance chemistry. And we've done it 40 plus times now. But as I'm sure, many folks on this Zoom know from bitter experience, solving one problem only reveals the problem behind it, right? And in this case, the problem behind it is no one really knows what a druggable site in RNA is. What I mean by that is that we don't know the rules for structure-function relationships in RNA, particularly in messenger RNA. We know it has structure, we know it has function, but we don't really know how those things line up. And I think we take for granted the fact that we have 60 years of structure-function protein biochemistry, so that when you recognize that a sequence is a protein kinase, right? Like exactly what you need to do. We just didn't know that with RNA, and so that is a thing we're now having to kind of figure out on the fly, and where probably a lot of our sort of secret sauce lies is recognizing where structure and function lines up, and in addition to that, taking advantage of our ability to find ligands more or less at will now, to in some cases skip the function step and say, you know what, all we need to do is find binders, and then we'll impose function on those binders. And that, for example, has led us to this targeted degrader collaboration with Amgen that we announced back in January.
So we've had a couple of questions about continuing on the M&A exit questions, but first we had another question as how you came up with the company name for Arrakis.
Yeah, well, it wasn't me, it was Jen. Prior to the movie coming out last year, what I found was that, half the people I talked to said, so what does that mean? And the other half, of course, knew exactly what it meant. Now most people know what it means, right? And so here is the metaphor. Arrakis is this inhospitable desert planet where no one really wants to go, except that it is the sole source of a critically valuable pharmaceutical. And because of that, the whole galaxy is fighting over it. And so better metaphor for RNA, I don't know.
And maybe that plays into your strategy for the exit M&A. So that's a nice tie-in if everyone's fighting over it.
There you go. Right, exactly.
So we actually did have a couple of questions. One is in general, sort of how the M&A environment has changed. And you all obviously announced the partnership with Amgen recently. And then secondarily, when you're thinking further along, the question is, how do you decide whether to, quote, exit early versus waiting for clinical development over time? And does the current funding environment factor into whatever that decision is?
Yeah, so I think to just answer the last question first, absolutely it does, right? Because if you can't get the capital, you can't advance your programs, right? That's sort of the extreme version. But I think we're going to see a lot of that, this year. But we sort of explicitly did that math at Padlock, right? Because we had a lot of confidence in the compounds we were developing, and we figured we knew exactly what we needed to do to prove their activity in the clinic. And so we explicitly did the math, which is to say, what would it cost us to take the program from where it is today through a phase 2A trial at RA? How long would that take? How much capital would we need to raise? At what approximate valuations would we raise that capital? And at the end of that, what would the valuation of the company need to be for today's shareholders to be indifferent, right? And the answer to that was something like $1 billion. And once we realized that, we're like, well, then, the fiduciary responsibility is you take the money today, right? Because that is a better return for your shareholders rather than making them wait 5 years with all the probability of success and all that sort of stuff. So that wasn't hard, actually.
Yep. So let's flip over to culture, because you mentioned the culture of Biogen and IDEC, and I think it's a little bit different in a startup because you obviously it's fewer people, it's a much more kind of compact team. And the question that we have is that you demonstrated really tremendous leadership in working with Jennifer Petter as she transitioned. And would you like to talk about kind of the challenges and leadership lessons from that experience?
Well, it's funny. I mean, I'm a little embarrassed by the credit I get for this because, it's not me. What was I supposed to do? Fire her? She sat me down at dinner one night and said, Mike, I'm trans and I'm in the process of transitioning. Here's the here's the date at which here's who I plan to tell and how and in what order, and here's when I'm going to start coming into work as Jennifer. And I'm like, wow, I was not expecting that. For me, really there were two considerations. I mean, one was to help Jen to the extent I could. I mean, this is a big deal for her. And I didn't want to make it any harder for her than it already was. And then secondly, make sure there's no impact, on the company. And it's my job to do that. I mean, my job as CEO to do the second thing. It's my job as a long-term 20-year friend of Jen to do the first thing, right? And and so we just did it. And I think that to me, what's probably so surprising is just actually how natural the whole thing felt. And how immediately accepting everybody was, and how we all just sort of rolled with the punches, and struggled a little bit with the he/she stuff for a few weeks and months. But it all feels quite it just feels like it always was. It feels like she was always Jen to us now. So
I think you get a lot of credit for this because you did it publicly, right? I think for things that are difficult to talk about and things, I think the standard company culture is, well, we talk about it in this room and we figure out how we're going to talk about it, and then maybe we'll tell somebody outside. And I'm sure you probably had that kind of discussion too, but outside of that, it was public, right? You blogged about it on the company website. I forget if Jen had a post on Bruce's blog or anything like that, but it was very much in the public sphere, in outlets that got a lot of eyeballs. And I think that's where a lot of the credit is, because I think that takes bravery to be that open and that forthright and that public about it. And I think that's really important, because I think there's a lot of other people whose issues and support, if you will, aren't that public. And I think that's actually probably a really, really big challenge for a lot of people with those considerations.
Well, maybe. I mean, Jen's the brave one. I was not the brave one. I was just doing my job,
Yeah.
I mean, the fact is, I'll just give you an example of, the kind of thing that we would have been just dealing with over and over again, right? As it was, we went to see an investor not long after Jen had transitioned, and halfway through the meeting, the investor said, "What happened to your previous CSO?"
Right.
And, And you know, the thing is, I mean, again, you know, I don't see what other choice I had, right? I mean, it was a very,
Yeah.
you know, I mean, it is a public thing, and you know, it's important, you know, important for the employees of Arrakis. It was important for the investors, present and future, you know, to know this was coming, and all of that. Anyhow,
Well, and
today
don't
it
you,
seems like a non-event,
I was
but
gonna say, yeah,
good.
and don't you think your proactive communication and Jen's plan made it clear that this was a normal process. It was something that was going to happen, and it's, you know, at the end of the day, it's good for everybody to be upfront and, you know, focused in what you're doing, which is developing the company, and not to let that be something that people got hung up on.
That's right. Look, I mean, you know, and Jen had been thinking about it for close to 60 years. I had 6 weeks, right?
Yes.
And so anyhow, I mean, it was really, I mean, it's a remarkable thing to go through. I mean, you know, you know, not just for Jen, but for everybody around, to be sort of up close and personal to that kind of, you know, that kind of an event. I mean, I'm not kidding when I say Jen's the brave one. Like, I just, you know, it's just hard to imagine what it was like to sort of, well, first of all, live that way for so long and not, and you know, and not take action, and then, you know, at this stage in her career and personal life, to do so. I mean, I just am full of admiration for her.
And and so how do you, let's take a step back. That was a very public, very specific example of ways that you had to set the team expectations and public expectations. How you've built a number of companies now, once you found your entrepreneurial calling. Talk to us about your decision process in developing your teams, because we obviously spend a lot of time in the New York ecosystem talking about building teams for early stage companies, and obviously it's different where you are in Massachusetts. But talk to us about your process.
Yeah, so, you know, I'll come back. I'll start with something I said about 15 or 20 minutes ago, which was recognizing that I needed to hire people who are a lot different from me. And, you know, and that's not, I mean, I don't think people come by that naturally, right? I think we, you know, certainly earlier in our careers, we tend to sort of hire in our own image. And you know, I've come to learn that that's exactly wrong. You need to hire people who, you know, who are highly complementary to you, right? Like if you know, if you're a big picture person, you need to pair up with a detail oriented person, right? You may drive each other crazy, but the fact is it's actually better for the organization to do that, right? And so, you know, I think that's really critical. I think the second thing that's really critical that is often overlooked is you actually have to interview for team chemistry. That, you know, when people go out to hire, they're often hiring to like plug a very specific technical gap in their team, right? And yes, you do need to plug that technical gap, but that is actually not the most important thing. And actually lots of people who can plug the technical gap, not so many people who will mesh well with the existing team. And so I think that's critical to interview for. And I don't have a magic formula for that, unfortunately. I mean, that's a little bit of intuition and judgment, and also listening to the other members of your team. And I've exercised that intuition on more than one occasion, you know, when I've been trying to choose between candidates where like one is clearly more technically qualified. I'm like, yeah, but I think this other one's going to be better.
Yeah.
You know, and anytime I've gone the other way, anytime I've overridden my gut judgment, I have regretted it. So I've learned to trust that.
Yeah. Do you do anything either specific or out of the box with the team or anything like that? Is there anything kind of non-standard that you do from an interview standpoint? I mean, there's, you know, you could have them sit down with the team, or is it really just that and you go by feel?
Yeah, no, we don't. I don't think we have any special, any special tactics that other people not use. I mean, we try to,
Yeah.
we try to ensure that individual interviewers are probing on sort of specific things.
Yeah.
I mean, the other thing that I think that's really critical is referencing and going past the references they offer and really finding people. And this is an advantage that I have, right? Like I'm so damn old now, and my network is so is so large that it's very rare for me to come across a candidate where I don't know somebody who's worked with me before,
Yeah.
right?
Yep.
And so, I could talk to somebody who will not lie to me, right?
Yes.
Mhm.
Yeah.
So
And
do you see, go ahead, Jennifer.
I was just going to say, so one thing that we spent a lot of time talking about too at New York Bio lately is increasing the diversity of our ecosystem, but also our networks. How do you go about looking for candidates that may not be in your immediate network?
Yeah, it's a good question. And I'm going to give you perhaps well, I don't know. Some people may like this answer. Some people may not. At Arrakis, we've actually had some pretty good success with external recruiters. Or an external recruiter that we've really worked with a couple of times, and it's been, who understands our organization and it's really been a good partner. And I was surprised by that because it started when we were, a couple of years ago, we were looking for a new head of chemistry, and we're like, oh, we know everybody, and Jen's view was like, I know everybody. And so, Jen, built a list of people she thought would be great for this role, and we kind of met all of them, and we're like, yeah, I mean, they're fine, but they're not really great. And so we ended up hiring an external recruiter, and they found us a candidate, a guy who's our CSO now, Jacques Dumas, who's fantastic and who was working like 3 miles away from here, but none of us had ever met or knew, because he'd worked in different sort of therapeutic areas, and just for whatever reason, our paths had not crossed,
Yep.
And so that was actually an important lesson for me, because I was pretty cocky that like there can't possibly be good chemists out there that I have to run into at some point.
Yeah.
So almost to kind of bring it back to the beginning, you had left Cold Spring Harbor to go to industry in an era that was very different in terms of the fluidity between academia and industry. And New York has, I think, the largest postdoc population in the world, and with the shrinking number of faculty positions, it's actually a really challenging choice between both industry and an academic track. So do you think about that when you either look at people coming out of academia? Do you think about kind of the challenges or benefits of going to industry versus trying to stay in academia, and how they deal with that choice? Do you have any kind of advice for younger scientists thinking about that?
Well, I mean, I think you're right that that there's just a real sort of bottleneck in the academic kind of career path. There's just not that many faculty positions available relative to the number of students and postdocs that are being churned out every year. And I don't think that problem's going to get any better.
No.
So I think I think if students and postdocs are being realistic, they should probably assume that industry is the default choice and not academics, right? And that's a complete switch, right? I mean, from what it was, certainly from what it was like in when I was in graduate school, right? Which was many decades ago, but and I think that's happening, right? I mean, we hire a lot of I would say maybe half of our scientific hires are first time industry hires, straight out of postdoc, or in some cases straight out of graduate school, and it's fun to have those folks here, and I think they have fun being here, and I think that I think that the challenging part of the transition for folks coming out of academics is one I referred to, almost an hour ago now, which is, recognizing that that you're no longer kind of a individual contributor, right? That you're working as part of a team. When you're in graduate school and you have a thesis project, I mean, you've got to go wherever that project, leads you. And if it requires application of a new method, then you learn how to do that thing, right? But here, if there's somebody else on the team who can do the next step better than you need to hand it off to them, right? And that's a little bit of an unnatural act for someone, coming out of an academic lab. But some people get it right away. Other people, it takes them a little while, but they get there eventually. I think. I mean, I obviously voted with my feet, almost 30 years ago.
Yeah.
I think, I think there's a lot of exciting stuff going on in our industry right now. Because it's a very competitive hiring environment, certainly, here in Boston, companies work hard to create a fun, attractive, and productive working environment for people as well. Like, as a postdoc, chances are your PI doesn't give a crap about, the quality of the space you're working in, or what the environment is like, or whether you're happy or whatever. We pay a lot of attention to, the health and well-being of our employees, because, we know they can get up and walk away at any time.
We only have a few minutes left. So use your crystal ball, and tell us what you see for your company in the next, say, 3 to 5 years.
Yeah. So, well, thank you for asking that question. Our operating assumption here is that there are 100 medicines that can be developed off our platform. I mean, although there's a lot of excitement in the industry over the last, say, 5-10 years, has been a sort of a string of new therapeutic modalities. The fact is that, and new biology as well. The fact is that something like 80 to 90% of human biology, kind of depending on how you do the math, is simply unavailable for medicine because a drug discovery toolkit can't lay hands on it. So, in my view, the most direct path to really creating value for patients, and therefore for all of our stakeholders, is by figuring out how to bring that biology into play. And that's what we're trying to do. And as I say, we think there's like 100 medicines here, and once you believe that, then a couple of things follow. One is like, well, we'll never develop 100 medicines ourselves, right? And so we should really deploy our platform as broadly as possible, with as many partners as possible, because at the end of the day, I literally don't care who sells these medicines to patients, whether it's us, or whether it's somebody else. Because at the end of the day, if we succeed in delivering all these medicines to patients, everyone will be fine, right?
Everybody wins. Yeah.
And so that's, 3 to 5 years from now, I hope there's, half dozen or dozen clinical programs, maybe 1 or 2 in our pipeline, but some in Roche's pipeline, some in Amgen's pipeline, and that people realize that actually you can drug RNA, and suddenly we've got the possibility of treatments for diseases that today are just off limits.
Well, I can't think of a better way to end the conversation today. Mike, thank you so much for your time. Thanks to everybody for participating. Have a good Tuesday, everybody.
Thanks, everybody.
Thanks, Mike. Thanks, everybody.
Thanks.
All right, see ya. Bye.
Thank you for tuning in to NewYorkBIO's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on NewYorkBIO, please visit us at www.newyorkbio.org.